Hi everyone. So the question here is, how do nerves stimulate muscles to contract? We'll talk about the properties of muscles, and in particular, we'll talk about how muscles are irritated by nerves at the neuromuscular junction. And as part of that, we'll discuss the sliding filament theory. Last of all, we'll talk about myasthenia gravis, which is a neuromuscular condition where the muscles are not able to respond to stimulation from the nerves. By the end of this lecture, you should be able to answer this question using all of these vocabulary. So muscles have two functional properties that are necessary for contraction. The first is they must be irritable, meaning they must be able to receive and respond to a nerve stimulus. And second of all, they must be contractile, which means that they must be able to shorten in response to a nerve stimulus. And when you talk about contractility, you're really talking about how the actin and myosin filaments slide against each other, which is described by the sliding filament theory. So we're going to discuss irritation for the most part, and that's how do nerves stimulate muscles? Well, skeletal muscles must be stimulated by a nerve. That's what allows your brain to control muscle contraction. And one of the ways that it's able to do that, or one of the main reasons why muscles respond in such a coordinated fashion to nerve stimulation is because muscles are organized into motor units. So a motor unit is basically one neuron and all of the muscle cells, which could be just a few to hundreds of muscle cells that are stimulated by that one neuron. So here you have a spinal cord, a cross-section of it. And then we have two neurons that are shown in this model. The red one, as you can see, has its cell body right here in this part of the spinal cord. So that's where you'll find the nucleus and all the organelles of this neuron. And then it has this long projection that goes through the nerve, and it connects with or synapses with this muscle fiber right here, this second muscle fiber, and the fifth muscle fiber over here that are part of this muscle bundle. You also see that there's a purple neuron here. Here's its cell body, and there's its long axon, its long extension, and this neuron synapses with or connects with the first, the third, and the fourth muscle fibers in this bundle. So that means we have two different motor units pictured here, the red neuron motor unit and the purple neuron's motor unit. And here you can see a microscope image of what that looks like. So when we talk about neurons, one thing you should just keep in mind is that it's a cell, but it's specialized. So this is a cell that you'll find in the brain, the spinal cord, and the nerves that are basically structured in a way that allows them to carry signals from one part of the body to another. So in this case, you can see that the signals are shown by the red arrows in this model, and they're coming in this direction down this long axon or this long extension where they synapse or connect with the muscle fibers right here at a region that we call the neuromuscular junction. And that's going to be on the next slide. I do want to just point out really quickly that we're able to now study neurons at a level that we weren't previously able to do using lots of cutting-edge techniques, one of which is optogenetics. So here, this is from a study at MIT, I think in 2017, where researchers figured out a way to cause individual neurons to light up in response to a particular signal they gave them. And so as we elucidate more of how neurons work, how they connect with other cells, what they control or what controls them, we'll have a better understanding of how to treat conditions like myasthenia gravis or other health conditions that have to do with neurons and what they control. Okay, so here we were just saying that the nerve ending right here of the neuron is going to synapse with or connect with muscle fibers at a region called the neuromuscular junction. So if we zoom in on there, here is the axon terminal, meaning the nerve ending, the end of that neuron, connecting right there with the sarcolemma, or the plasma membrane, of one muscle fiber. And if you zoom in on the place where they connect, it's going to look like this. Once again, here is the nerve ending of that neuron, the axon terminal. Here is the plasma membrane of the muscle fiber. It's called the sarcolemma. And then one thing to notice is that there's a gap between these two. And that's significant because it allows a level of control that that you get when a signal tries to travel from the nerve to the muscle. So in order for that signal to be transferred from the nerve to the muscle, the signal has to cross this gap. And the only way to do that is to use these chemical messengers called neurotransmitters. And the only type of neurotransmitter you need to know about for this particular unit is acetylcholine. But basically, this region right here is called the neuromuscular junction, and this gap right there is called the synaptic cleft. And a signal from the brain that's traveling down the nerve must cross that gap by using these molecules shown here in these vesicles. So how does that actually happen? Like, how does a nerve signal actually trigger a muscle to contract at the neuromuscular junction? So let's start with B right here in this model. You can see that this is the action potential, so that's that flash of light that's traveling down this axon terminal of this nerve. And what happens is the action potential that comes from the brain, or that originates from the brain, travels down this axon until it reaches the neuromuscular junction right here, where, now we're at A, we're at the nerve ending, and the nerve ending right here, this axon terminal, is going to release these little yellow dots, these little yellow chemicals called acetylcholine. And we abbreviate that with the letters A, C, H, capital A, capital C, lowercase h. So acetylcholine is a special chemical messenger that is released right here between the nerve ending and the muscle fiber. And that's going to then cause that's going to allow the acetylcholine molecules to cross that gap and bind to receptor proteins on the muscle fiber, so on the sarcolemma membrane of the muscle fiber. So when that happens, when the acetylcholine chemical messengers bind to receptors on the other side, that's going to trigger the entire sarcolemma membrane to depolarize. So you can see there's this flash of light that's going to travel through the sarcolemma membrane in all directions. And it's also going to travel down these T tubules. And so that's what you see here with letter D. So these T tubules basically invade the interior of the cell and allow the action potential to not only travel along the outside of the muscle fiber, but deep into the interior of the muscle fiber as well. And so what happens when the action potential travels deep inside? Well, then the action potential travels through the sarcoplasmic reticulum, which, remember, is a specialized endoplasmic reticulum in muscle fibers that stores lots of calcium ions. And so you can see that flash of light is now going to travel down the T tubules through the sarcoplasmic reticulum, which is shown to you here with letter E. And that triggers the sarcoplasmic reticulum to release calcium ions, which are those little white dots that are shown or labeled with the letter F. So now calcium ions are going to flood this entire sarcomere here, and the next sarcomere, and the next sarcomere. So all the sarcomeres along the entire length of the muscle fiber are flooded with calcium. At the same time, you have this mitochondrion here that is converting ADP, which is shown in orange, into ATP, which is shown in, I guess, a lighter orange. And so now you have all of these mitochondria everywhere throughout the entire muscle fiber, just producing tons of ATP. And the ATP and the calcium are now both flooding the entire length of the muscle fiber, where they trigger the myosin filaments, which are shown right here with letter H, and the actin filaments, which are shown in magenta, letter G. They trigger the myosin and the actin to bond to each other and form what are called cross bridges or connections between the myosin and actin filaments. And that allows the myosin to pull the actin towards the center of the sarcomere. So they slide against each other, and that's what we call sliding filament theory. So as the thick myosin filaments and the thin actin filaments, shown here in red and in magenta, as they slide against each other, what's happening is that causes these Z discs, which you can see over here on the left boundary of the sarcomere and on this right boundary of the sarcomere. It causes the Z discs to get pulled in closer towards the M line in the center, which means that the H zone, which is this area in the middle that is missing myosin, the H zone gets narrowed as the entire sarcomere contracts. And you can tell that the entire sarcomere is contracting because, once again, look at the Z discs. They're coming closer together. And look at the H zone, it's narrowing down. And when the entire sarcomere shortens, all along the length of the muscle fiber, when they all shorten together, that's called contraction. And so the end result of that is smooth movement. So here you can see you've got the adductor muscles in your thigh. And right now they're relaxed, but as they shorten, as they contract, and as these muscles shorten in length, they pull the leg towards the midline. So they adduct your leg. So how do sarcomeres contract? This is described by a theory called the sliding filament theory, and it's the idea that when you have calcium and ATP present, that's going to allow the myosin heads right here on this thick myosin filament to bond to the actin right here on this thin actin filament, and to pull the actin towards the middle and then to release. So you can see this one pulls and then releases and then binds again. If you want to get into the nitty-gritty, this is more of like honors level material, and so for my class at Pacifica, this particular slide would be optional. But if you want to get into the nitty-gritty, this GIF is showing you how it's done. So here you have a calcium ion binding onto the actin filament and basically changing the shape of these ropes that are on it so that it exposes the actin and lets the myosin bind to it. Remember, the myosin heads can only bind to the actin if calcium is present. And then that allows the myosin to bind and to pull the actin towards the center of the sarcomere, and then they use ATP to release and bind again. Okay, so now this part, you should be able to just pause the video at this point and take a minute and see if you can walk yourself through what is going on in this model as you zoom in from the larger picture here to the more detailed pictures down here. And then when you're ready, you can unpause. All right, so everyone should be able to do this in this class. The idea is an action potential, which is a signal from the brain, is going to come down this neuron where it's going to connect with the muscle fiber that's part of the motor unit for this neuron. Remember, a motor unit is one neuron and all of the muscle fibers that are part of it. And so this muscle fiber, if we zoom in right here at the connection between the neuron and the muscle fiber, that's called the neuromuscular junction. It's the junction between a neuron and a muscle. And so here we can see that there's a gap between the end of the nerve and the beginning of the muscle fiber. And so if we zoom in on that gap right there, this is what we see. We see that the nerve ending, shown in yellow up here, is releasing acetylcholine molecules, which are chemical signals that then diffuse across that gap and bind to these purple receptor proteins on the muscle fiber's sarcolemma, which then triggers an action potential to travel throughout the entire muscle fiber. One side note, just keep in mind that nerves stimulate sarcomere contraction in the way that like a match will trigger an entire twig to go up in flames. And so if the match is not, like if the fire is not strong enough in the beginning, the twig won't catch on fire. But if it is strong enough, then the fire will catch and it will travel all the way down the length of the twig. And once you start it, it's pretty much impossible to stop the traveling of that fire. In a similar way, here you have an action potential coming from the nerve fiber. If that action potential is not strong enough, you will not get a muscle contraction. But if it is strong enough, and in particular, if you have multiple action potentials that are coming down repeatedly down the nerve fiber and adding up together, then that can trigger a muscle contraction that immediately will spread automatically and almost instantaneously down the entire length of the muscle fiber, which allows all of the sarcomeres to then contract at the same time. In the meantime, you have enzymes present in this gap here, as well as possibly present in the presynaptic membrane, in the nerve ending. But you have these enzymes that are present, and their job is to break down any of these chemical messengers, these green acetylcholine molecules that are left after the action potential has been started in the muscle. Here's why. If you let acetylcholine molecules just kind of linger at the neuromuscular junction, then they could continue to bind to receptor proteins on the other side, which means they could continue to trigger muscle contractions one after another, even if you didn't intend for that to happen. And so those are muscle spasms. That's where you do not have full control over your muscle contraction. So that is why one nerve impulse, only one action potential traveling down the nerve, will produce only one muscle contraction instead of continual contraction. So that allows your brain to better control your energy and your movement. Okay, so why don't you take a minute, pause the video, and just see if you can explain to yourself, how do nerves stimulate sarcomere contraction? Okay, and when you're ready, you can unpause the video. So once again, an action potential or a nerve impulse is going to travel down this neuron, where it's going to trigger acetylcholine chemical messengers to be released right there at the neuromuscular junction. That's going to let the acetylcholine molecules bind to receptors in the sarcolemma membrane on the other side, triggering an action potential or a new signal that's going to travel down the entire length of the sarcolemma and down the T tubules into the sarcoplasmic reticulum, triggering the release of calcium. So the sarcoplasmic reticulum releases calcium, which then floods the cell at the same time that ATP is flooding the muscle cells from the mitochondria. The presence of calcium in ATP then allows the myosin filaments, the thick filaments shown here in red, to bind to and pull in the actin filaments, which are shown here in purple or in magenta, which then causes the thick and thin filaments to slide against each other, shortening the entire sarcomere. And that's a muscle contraction. Last of all, let's discuss myasthenia gravis, which is a chronic autoimmune neuromuscular disease, which means that your own immune system, if you have this disease, is attacking your own body. So what's happening? Well, you're going to lose control of the skeletal, which means the voluntary muscles in your body. And you're going to have varying degrees of weakness in all your skeletal muscles, beginning with weakness in your head and your neck muscles. And so one early characteristic is droopy eyelids. Another is difficulty chewing because you lose control of the masseter muscle in your jaw. Another is you lose the ability to swallow easily or to talk clearly, and you may eventually end up with difficulty breathing. So why does this happen? Why are you losing control of your eye muscles, your jaw muscles? It's because you're going to have fewer acetylcholine receptors that are available on your muscle fibers. And the reason why is because antibodies from your white blood cells will bind to the acetylcholine receptors because your body thinks that those receptors are foreign invaders. So it produces antibodies to try to fight them off, but what you're really doing is you're destroying receptor proteins in your muscles that are supposed to be there, that are supposed to bind acetylcholine. So because the receptors are not triggered, that leads to weakened muscle contraction. So here in the schematic, in the top panel, we have a normal neuromuscular junction. Here is the nerve ending with all of the acetylcholine inside. Here is the muscle fiber sarcolemma, and you can see that it's folded in on itself to increase the surface area so that you have the ability to pack even more of these acetylcholine receptors in there. The more acetylcholine receptors that are there, the more easily acetylcholine can bind and trigger a strong action potential. The stronger the action potential that you trigger, the stronger and smoother your muscle response will be. But if you have myasthenia gravis, look at the bottom panel. Because your immune system is attacking the acetylcholine receptors, here you have these antibodies, these blue antibodies produced by your immune system cells, which are binding to the acetylcholine receptors at your muscle fibers, which inactivates them. That then causes the muscle cell to destroy the acetylcholine receptors because of homeostatic loops. So that means that over time, your muscle fibers degrade. They can no longer respond to acetylcholine, and that essentially weakens and eventually destroys their function. So on that sad note, here are your key takeaways. This was a really conceptually dense topic, and so you may need to go back and rewatch parts of this video again, or go look for other videos, or read into your book to make sure that you master this concept. But here are your four takeaways. Four-ish takeaways. So the skeletal muscles must be both irritable, meaning they can respond to stimulus from nerve cells, and they must be contractile. They should be able to shorten in response to that stimulus. Second, action potentials traveling down neurons irritate the muscle fibers of the motor unit for that neuron by depolarizing every muscle fiber at its neuromuscular junction. So how does it do that? Well, that's described by the sliding filament theory, which describes how basically calcium ions and ATP can cause actin and myosin filaments to form cross bridges or connections with each other, which means they slide against each other and shorten the sarcomere. Next, you need to know that each action potential triggers one muscle contraction, and that myasthenia gravis is a disorder in which the body produces antibodies that attack the body's own acetylcholine receptors, which means that your muscle cells are no longer able to respond to nerve signals, and that weakens muscle contractions. So once again, this was a really dense topic, and if you can answer the essential question that was at the top of this, this presentation, using all of the vocabulary that are here, then that means that you have mastered this topic.